our speaker today is Dr. Beskok, and he is very accomplished. He received his BS in mechanical engineering from the Middle East Technical University in 1988, and then he received a master's degree in mechanical engineering from Indiana University, Purdue University in Indianapolis in 1991, and then he got another master's and a PhD from Princeton in mechanical and aerospace engineering in 1994 and 1996, respectively. He was a visiting scholar at Brown University Center for Fluid Mechanics from 1994 to 1996, and then he was a postdoctoral research associate at MIT, specifically the Research Laboratory of Electronics, from 1996 to 1998. And then he went to Texas A&M in the mechanical engineering department as an assistant professor in 2007, he moved to Old Dominion University Mechanical and Aerospace Engineering Department as the Batten Endowed Chair Professor of Computation, Director of the ODU Institute of Micro and Nanotechnology. And then in 2013, he finally came to SMU as the chair of the ME department, and he served in that capacity until 2019. And currently, he is the Brown Foundation Professor of Engineering at SMU. So, over to you, Dr. Bauer.
Thank you, Casey. So apparently, of course, I got all my degrees before you were all born, which is kind of makes me, wonder about it. Years ago, when people introduced me, I wasn't as old. But anyhow, thank you very much for this kind invitation indeed. Today I'm going to introduce you to the research we're doing in our laboratory, which is in Embry building, second floor. We are basically building a microfluidic device that is to be used for detecting COVID-19 antibodies, right? So what I would like to do is actually just get you filled in with, some very recent exposure that we had, so that these are very small movies I will show, and then I'm going to tell you more about what is going on, all right? So the first one is actually an elevator pitch video that we created, and we created this in, April 2020, as we were trying to hit some funding. So here we go.
Let's face it, the COVID-19 pandemic is ravaging communities and crippling the world economy. Many nations imposed stay-at-home orders for the general public. Health officials are uncertain about when to lift these orders and how soon daily life can return to normal, determining the actual spread of COVID-19 infection, which requires extensive testing of the population. However, testing large portions of the population requires the development of quantifiable, accurate, fast, and inexpensive diagnostic methods based on detection of human antibodies, which detection is ELISA, which is expensive, slow, and hard to scale for testing large populations. The common lateral flow assay are fast, but not reliable. Fortunately, researchers at Southern Methodist University developed a lab-on-a-chip concept to address these very needs. The portable system will use 1 milliliter of blood, which will pass through a filter layer to obtain plasma. Then, the chip will be put in a portable unit that uses electrical measurements for detection of specific antibodies. The results will be wirelessly transmitted to a smartphone interface that will display accurate, quantitative results. The proposed device is based on our preliminary results shown on the right. We were able to detect IgG antibodies at 1 nanogram per milliliter level within a minute. The research team consists of doctors Beskok and Chiao. 50 distinguished experts in the field. The device can be modified to detect other diseases, such as tuberculosis and malaria. These will extend the impact of our technology to diverse and larger markets. So please, help us shape the future of antibody detection with our unparalleled technology so that we can truly understand the extent of this pandemic and help health officials make informed decisions.
So I just want to show you another 2-minute video, and this is happening due to a young lady came and interviewed us, and she's from SMU's TV, and I didn't know that SMU had a TV. So here we go. Excuse me.
Hey Mustangs, I'm Lauren Rangel and welcome back to Coronavirus on the Hilltop. This very moment, health officials are distributing COVID-19 vaccines, but there is still so much left to learn about the pandemic. A team in the Lyle School of Engineering is developing a way to help. Here's more.
We want to change the world, and this project is a perfect demonstration. How do we work on something that is direly needed for our society?
A team of researchers at SMU are developing a new COVID-19 antibody test called Lab on a Chip. All it takes is 1 millimeter of blood. A filter inside the chip extracts the blood's plasma. Then the chip goes into a portal unit that uses electrical measurements to detect antibodies. Even with a vaccine in progress, Lab on a Chip is a game changer.
We still know very little about the effectiveness of vaccine. How long the antibody lasts? We need to know before we can stop this pandemic.
There are other antibody tests available, but they're either too slow or not accurate enough.
The other methods take 3 or 4 hours to give you the results, but this method gives you the results.
Simple and easy. It could be available to the mass.
Our and everybody can have this instrument.
But this will happen without funding.
To be successful, we need institutional support and necessary infrastructure to go forward.
In a once-in-a-century pandemic, the stakes are high.
People are being hurt. We can actually help people and impact humanity with this.
For now, they're focused on living up to the SMU slogan, World Changers Shape Here. It's COVID-19. It works with other diseases like tuberculosis and shows promise for future pandemics. That's this week on the show. Feel free to send them to @Lauren_K_Ray.
All right. So I guess this really tells you, a little bit about what I'm going to talk about, right? So you have seen these videos, and the key thing is that now most of us are getting vaccinated, which is great. However, there's a problem. We do not know how long our antibodies are going to last. If you look into our flu influenza vaccines, we have to get here is going to be a different strain. And we know that COVID has different strains as well. And we don't even know whether the current vaccinations are good for or as effective for all those strains. Regardless, everybody's getting vaccinated, and it by itself is a big deal. I think 385 million Americans, and hopefully most of them will get vaccinated. Imagine the fact that we don't know how long this is going to last. Is it 3 months, or is it 6 months, or is it 9 months, a year, or maybe for life? We're saved. We don't know, which means that we have another problem coming ahead of us. So basically, we have to keep on testing people to make sure that the vaccines are still relevant, and we have built enough antibodies to withstand COVID-19 3 months, 6 months, a year, whatever down the road, right? So this actually is another challenge, because it is very challenging to vaccinate everybody, And imagine trying to measure the antibody levels on people, very frequently. It's a big deal. It's a big challenge. So what is the key thing is that, let me actually get the laser pointer here. So the primary technique to do this is called enzyme-linked immunosorbent assay, which is ELISA. It's a plate-based assay technique that detects in a quantifiable fashion soluble substances like antibodies. So the key is that the substrate is going to have an antigen on it, and for COVID-19 is going to be the spike proteins. So the spike proteins will, when a primary antibody, say from your blood plasma comes in, will attach to it, because these antibodies are for these COVID-19 spike proteins. So, however, you don't know which antibody is attaching. Is it IgG, IgA, IgM? And if you would like to actually know which one is really attaching, you have to put a secondary antibody as a conjugate. And if this is IgG, it will only attach if there is an IgG in here. All right. So IgG is going to be an antibody that we require for long-term defense against, COVID-19. IgM is a very fast one. It basically peaks, and then after IgG is produced, maybe in a week or two, it just basically decays. IgA is an antibody for mucosal, tissues and so on. And of course, it's related with, if it is coming through your throat or nose, those things will be actuated. So the bottom line is that you need to do this ELISA test, which will require 2 stages. Dealing with an antigen on the substrate that catches in a non-specific way the primary antibodies, and the specific reaction will happen when you send in the specific secondary antibody that's the conjugate of what you're trying to catch. When that happens, you're going to have an optical reaction. You're going to see a change of color, and it tells you that you have actually certain antibody in your blood plasma. Now, this is the state of the art, and in biological sciences, it's very difficult to let people accept new technologies. Once there's a gold standard, people will stick with it. But this current gold standard ELISA will not actually allow us to test maybe 385 million Americans maybe 3 times a year. It's not possible. It's too expensive. So it's not portable, expensive to purchase, expensive to operate. You need trained personnel to actually work on this. And for those of you who are engineers, the whole detection based on diffusion. So basically, if I have, for example, a droplet of dye in a pool, and the dye will be concentrated, if you really wait for a long time, that dye is going to basically diffuse out. And there will be little dye particles here and there, but say in a pool, it may take maybe a year or so for a droplet of dye to totally disperse only with pure diffusion. So for those of us who are engineers, we would like to make it happen better by inducing mixing and convection. Mixing and convection will make things rapid, and hence you can actually beat the time scales for diffusion. And also the detection that they use is optical, of course, well accepted norms. So although that's the case, starting from early 2000s, basically Professor Whiteside from Harvard University coined the term laboratory on a chip. He is a chemist, so he's an analytical chemist, and he figured out that perhaps we can actually reduce the whole chemistry laboratory onto a specific handheld device that will do 1 or 2 things for you, not like 20 different things. Obviously, a large chemical lab will be able to handle a lot of different things, but assume that you would like to focus on a couple of markers, couple of diseases, then it is possible to actually build these, lab on a chip miniaturized devices. This is very interesting because it's come from New York Times. At that point, they did not coin the term lab on a chip. Instead, it says tiny pipes offer big payoffs. We don't have exactly pipes, we have channels. But anyhow, so the key is that can we do this faster, cheaper, and better? And can we actually build, portable units that can be used, all together by many different people? So you can actually test, immunity with respect to versus diseases, certain diseases, using these kind of devices. So I'm going to tell you a little bit about the device that we made. So we use basically a microfabrication technique to sputter some gold onto gold glass surfaces. And here's the picture here. If you would like to zoom into this, there are basically 3 dark spots. Those dark lines are actually gold sputter. And this is typically 20 nanometers thick. And this is about 200 micron by 200 micron opening. Here is the 3rd floating electrode, and here is actually another electrode. So we're going to use these electrodes, and here's a picture of how we're going to be energizing these electrodes. So these are planar electrodes. But what we do is that we use actually gold coating technique, and we use an alumina, which is aluminum oxide membrane, as a stencil. If you take an aluminum foil and if you properly oxidize it, you're going to actually get, cylindrical structures inside an alumina membrane. We use this as a stencil, and we build so-called, nano rods. We build rods, and I'm going to tell you about them. By doing that, I'm increasing the surface area. Surface area increase means you can actually catch, more antibody within the same sensor. So let's actually talk about length scales, because in our daily lives, we're not used to seeing microns and nanometers. So micron is a millionth of a meter. It's very, very tiny. Nanometer is 1 billionth of a meter. But guess what? We all actually experience micro effects, in our lives. So if you actually pick a hair from your head, more than likely it's going to be about 75 to 50 microns thick. And this is the scan electron microscope picture of human hair. So this thing is about 50 to 70, 75 micrometers. Another thing in our lives that's about maybe 100, 125 micrometer is basically the paper, a thickness of a paper, all right? So these are the things that we see, but not as common. So we take this and we basically create these electrodes, and then using the gold deposition techniques, we create a bunch of nanorods. Look at this scan electron microscope picture. I cannot tell the scale. This is 1 micron, okay? Look at in 1 micron, I have a bunch of little tiny dots. Let's see what those dots are. So with 200 nanometer resolution, looking from top, there are a bunch of, like a comb. It just comes up, all right? Many, many nanorods. And here's another picture. And this is 100 nanometers. So these fellows are about 80 nanometers in diameter. So what we do here is you tilt your sample, you take another SEM picture. Now you can actually see more of them, all right? So typically we have about 200 nanometer height with an 80 nanometer diameter, and we have billions and maybe more than billions of them on this surface. And we could actually build these, with the patterning of the electrodes that we have here. This makes it a very unique surface. The surface area enlarges. It enlarges by a factor of 7, all right? So although you're dealing with maybe 200 micron here, and that length is like 1 millimeter, that area is, of that surface structures that we put in. I would like to put the surface structures so that I can cover those surface structures with my antigens, all right? And hence the antibodies come and attach. That's an idea. On the other hand, what I try to do is, although this is gold when I'm going to put these, nanorods, I'm also going to make the glass surface hydrophobic. What does hydrophobic means? Not liking water. So the glass surfaces is not wettable by aquatic samples, aqueous, samples. And, my nanorods are covered with antigens, all right? So This is the whole idea. So if you look into a picture here, okay, and obviously this is a dry picture, but when we put HMDS, which makes this hydrophobic, you put a droplet on it, those droplets are not spilling through. If you put this in a regular, gold-coated surface, it will have a wetting angle that's going to make it, more hydrophilic, which means liking water. So the droplet is going to spread over the gold. However, you put the, droplet over HMDS, it doesn't like it. It basically creates a large angle of contact. So this is very important, all right? Although having hydrophobic surfaces is super hydrophilic. We basically create these nanorods, we expose it to UV light for a certain time frame, and then the water droplet just covers the entire structure of the electrodes. But the rest of the channel is hydrophobic. So then what happens is that we can actually put tiny little dots of antigens to functionalize these electrodes, and you can do it with a pipette, dot by dot, and you will be able to, of course, I'm functionalizing the entire surface, all right? But the rest of the channel, which is going to be passed through here, is hydrophobic. Hydrophobic means that these antibodies that I'm going to introduce afterwards would not like to get attached to the surface because they are not really touching the surface, all right? So this is basically an idea that we had, and this basically minimizes the amount of antigen that you use. So basically, these antigens are extremely expensive, these spike proteins for COVID-19. And by doing this, we minimize the use of the antigen, because if you put a droplet that just goes everywhere, you know what's going to happen? That droplet is going to be functionalizing the entire channel, which means that your antibodies can attach anywhere. But by doing the channel hydrophobic, I make sure that my antibodies basically attach only on the electrode surfaces, all right? So that is actually one of the intellectual property we have in our patent application. The other one is how to beat diffusion, all right? So you have usually a high ionic conductivity, blood plasma, and that's about 1.5 Siemens per meter is its value. So this is highly conductive. So if you put a conductive fluid and you apply electric fields, what do you have? Anyone? You have Joule heating. Joule heating is something you cannot avoid, but in this case, we design it such that it is helpful for us. So we induce through Joule heating something known as AC, which is the AC frequency, electrothermal flow. So what really happens is, above my electrodes, I'm actually having the fluid to turn around. So it's coming directly in and hitting, the whole idea is to hit here and gather all my, if this is really seeded with antibodies, it's going to attach everywhere on my sensor region. That's the idea. But this is what we call convection. Why convection is important? It is important because it beats diffusion. So if you actually take an electrode here, and this is the numerical simulation of the actual process of, grabbing a target initially at 1 nanomolar concentration. If you wait for basically 60 seconds, you will see that only regions that are very close to my capture electrodes are going to feel the capture. But because of the fact that I'm inducing convection, the electrodes are capturing more and more of the antibodies as a function of time. So that's the beauty of using electrothermal flow. If you would like to do this application in low conductivity buffers, then you have a similar sister effect, which is known as AC electroosmotic flow, and it does very similar patterns as well. So the beauty of this is this design can make it fast capture for environmental samples, which will have lower conductivities using AC electroosmosis, but for physiological conditions with high ionic conductivity, you can actually capture, with AC electrothermal flows. So going forward, you can actually see the device that's put together. We actually use a double-sided tape here to create a channel. We drill the holes from the top surface. So there's an inlet, there's an outlet. Double-sided tape is about 150 microns high. And of course, the whole channel is hydrophobic, except from this region where we have hydrophilic. So if you put a droplet somewhere here, it just runs away. But if you put a droplet where we actually put, of course, our antigen or the spike proteins, it creates this droplet. So, and of course, the surface is full of nanorods and increases the surface area. So here is actually the actual experimental result. And we have received this, we have this result from 2020. We used at that point bovine IgG, whole molecules, and we would like to capture them. So in order to look at the capture, let me tell you how we're doing the capture. Because we're applying a certain electric field to create this flow patterns, we do these at 5 megahertz excitation, at 4 volts peak-to-peak, excitation on these electrodes. It creates these kind of flow patterns when you apply 4 volts. Then if you switch and you say, this basically function generator creates the 4 volt peak-to-peak, and then we are measuring the electrical impedance. For those of you who are mechanical engineers or non-EE, electrical impedance is a complex number that's a combination of the resistance as well as inductance and capacitance of the system. So you're measuring this a complex quantity. This complex quantity becomes highly resistive in high frequency. Highly resistive means mostly real. Real means you get resistance, and you measure this at 5 megahertz. So we are actually actuating our electrodes at 5 megahertz, at 4 volts peak to peak, and we are measuring the impedance response again at the same frequency, but this time like 0.01, like 0.01 volts peak to peak. The beauty of this is this: the actuation frequency is the same as the measurement frequency. Although I have lab grade humongous devices here, I can easily, with the help of course, Dr. J.C. Chiao, I can't do that, but he's an electrical engineer, he can. We are trying to actually replace this equipment with dedicated circuitry, right? So a beautiful way of actuate at the same frequency as you measure, which makes it very easy to do this electronics. The only thing is changing is the actuation voltage versus measurement voltage. Having said that, now let's look into the results. So this part on the left are the results that we have obtained by using gold planar electrodes. There are no nanorods on them. The results on the right has nanorods. So if you actually would like to measure the impedance as a function of time on this chip, after you put in plasma that contains bovine IgG, you will see that without actuation, nothing is really changing. What we're looking at is the change in the impedance at 5 megahertz divided by the original impedance that we had in the chip before we actually put the sample that has IgGs in. So the diffusion is so slow, it will pick up maybe in an hour or so, we're going to see a response. This is the deficiency of ELISA. By minimizing our chamber volume, we are able to actually, and employing the convection, we are able to actually get much faster results. So in the planar electrodes, let's look into a case. We have 100 nanogram per ml, and here is the response for it. The red one is 1 nanogram per ml, and the blue one is, oh, I'm sorry what the bias is, but Here's 100 nanogram per ml, and the blue is 10, and the red is 1, all right? Let's do the same experiment with nanorods. Diffusion is still very slow, but even with 1 nanogram per ml, which is actually a very reasonable amount to detect for immunity assessment, we are able to get about 20% response in the impedance change in our chip. And of course, it takes a little bit longer than the higher concentration. So currently, we are working on extending this data to longer times, and we're also trying to extend this data into lower levels of detection. So I'm sure that if I can capture 1 nanogram per ml with a great signal, probably I can do 0.1 or 0.01 nanogram per ml. If I can quantify the data as a function of the concentration of the antibody, then I can actually say in your blood plasma, this is the level. And that's beautiful because that's what you need. And there's a threshold level that's going to tell you that your immune or your immune system will basically fight against COVID-19 if it gets into your system. All right? So this is the idea. So as you have seen, we have used a lot of electronic equipments, and these are maybe $5,000 is just a function generator. Impedance analyzer brand new is $50K. Well, those have to be actually shrinking into a handheld device size so that this idea would work. That's why I'm working with Professor Chiao. So as you have seen in the movies I've shown earlier, you have a drop of blood that goes through a plasma filter layer. It gets rid of your white and red blood cells, and the plasma enters the chip. And you plug this into an interface module, which is going to do the measurements. And using Bluetooth, it's going to tell to your smartphone the levels that it is measuring. At the same time, it's going to upload the data in the cloud so that people like health care officers and your doctor would know whether you have a booster shot or not. All right. So this is the idea that we have. So so let's actually talk a little bit about things that we haven't really talked about this device. All right. So the whole idea is that I was going to start with spike protein, and when your blood plasma comes in, all different kinds of COVID-19 antibodies in your system will attach to it, because all of them are identifying it as spike protein that belongs to COVID-19. For that reason, I must have a secondary, so this is a general way of, or generic way of capturing everything related with COVID-19, then I must use a secondary specific antibody. So our device basically is not a single channel device. It is a device that has radially going outward channels. You can have maybe 4, if you want 15 or 20 different channels radially going out. So the idea is simple. After I put spike proteins onto these sensors, and the sensors are all connected to their respective measurement system, all you need is one impedance measurement that basically switches from sensor to sensor in reality. All right, so it's easy as an engineering. But you put your blood plasma and you push it into this channel. You push it so that there's still a gap between the secondary reservoirs that contain different specific antibodies like IgM, IgG, or IgA. So what happens at that point is use AC electrothermal flow, generically capture everything related with COVID-19 antibodies. So your impedance will increase. And I'm hoping that they're all going to increase equally, because we're all capturing everything related with the COVID-19 antibodies from the same inlet. So it's the same sample. Then what you do is you can build this reservoir so that as you push down, it's going to actually let a secondary specific antibody comes in, and then it fills the channel and pushes the sample out into the inlet. And when you apply AC electrothermal flow, now you're actually measuring what you have Is it IgM? Is it IgG? Is it IgA? So this is how the device actually works. So far, we have shown that within a few seconds, in 60 seconds, we can do the first process, which was a general capture, but our data used only IgG. So it was really capturing IgG, not every antibody related with that particular system. But our sensitivity was 1 nanogram per ml. So by using multiple species, by using this single inlet and radially out design, we don't have cross-contamination of our sample. So we can actually detect n different species from n different channels. So what we have is really flexible. It doesn't have to be a COVID-19 device. It can be for malaria or tuberculosis or anything else that we want. So it can be changed that way, all right? So as I said earlier, I'm using AC electrothermal flow, which is suitable for physiological buffers that have high conductivities. But if I use low conductivity buffers, just say like you get a droplet of water from the lake, or you have a drinking water, and their conductivity will be much less. But you will still see that this mixing due to convection works well, because then you will use AC electroosmotic flow. So in addition, we use hybrid surfaces. Our channel surfaces are totally hydrophobic, so that it will reduce basically the friction that happens as the flow is getting in and out. But also it reduces non-specific binding, because the antibodies cannot get close to the surface and unspecifically attach. So that's a good thing to have. But of course, our electrodes are ultra hydrophilic, and that's where we capture most of everything. And the last thing that we have really talked about is that by using nanostructured electrodes with nanorods, we tremendously increasing the binding surface area, which allows also reduction of what we call electrode polarization effects. So as you can see, this is really a nice package deal. We have done the single channel version of it, and we are hoping to get into this 4-channel version. Currently, we have a seed grant from Lyle School, but I really would like to actually get something bigger and hopefully work with UT Southwestern partners as well. And without them being involved, there's not going to be a large NIH project. So the sooner we bring them in, hopefully we'll be able to hit NIH funding. And I really hope that there will be companies who are interested in this technology as well. So we are basically working towards this direction, right? So that's it, folks. It took about 40 minutes with 10 slides. Any specific questions?
What would you
go ahead?
You can go.
No, go ahead, Mia.
What would you recommend for an undergraduate who wants to get involved in biomedical research? How do you think that we should go about that?
That's a very good question, Mia, because you said that you're a mechanical engineering major doing biomedical. So actually, let me be very honest, the biomedical program is identical to the Deadman's biomedical program, which is geared towards you taking your MCAT, exams. So that actually is not exactly what I will call a biomedical program, or bioengineering at least, right? It's really biomedical perhaps, but there's not an engineering to it. So when I was a department chair, and I was one till 2018, or was it 19? Oh my God, it's 19 actually. All right, so not bad. So see, like I forgot those days. It wasn't easy. But the whole idea was to work with the electrical engineering department to initiate a bioengineering related, under a separate name, maybe bioinstrumentation master's program, and make this as a 4+1 program. So as you go take all these courses for MCAT, we really hope that we could actually really tell you engineering aspects of it. But regarding to what you need to do, if you have time, please, of course, first dedicate everything to academics because you must pass and graduate. But if you have time, identify a professor that you know personally, taken their class, or you have met for some reason. Talk to that person and tell the professor of your interest, all right? And we love to have undergrads in our lab. But there is really an interesting catch here. I have done this since I came to SMU, but some undergrad says, oh, I want to do research with you. They come and stay for a week, and then they're gone. So the time that we spend with these students, because we need to really get them through a lot of training, they get bored at the training phase and they're gone. On the other hand, I had three ladies, all of them are mechanical engineers, stick with me. One of them actually wrote 3 journal articles and ended up in an bioengineering work through UT Southwestern. Another one basically ended up in Texas Tech Medical School. And what happened to the other one, which I don't know what happened to her. But anyhow, so basically, people who stick with it, this kind of research takes you somewhere, all right? And the person who went to Texas Tech actually co-authored the patent with me. So, and of course, I'm talking about my own experiences, but Lyle School of Engineering has plenty of faculty who is doing different aspects of biomedical research, both for MEs and ECs. Now, for example, from the ME side, if you're interested in dynamics and controls and human locomotion, Professor Hürmüzlü or Professor Edmund Ricker would be the right people to talk to, all right? If you're interested in, for example, DNA transport through nanochannels, Professor Minjun Kim does that. He also let virus particles try to pass through nanochannels, where he's trying to measure the mechanical properties of these viruses. So he's also building microscale robots that can one day, using magnetic fields, travel in your veins, all right? So that's the thing that what Minjun does. I talked about what I do. I also work on another project that is used to identify the electric properties of cancer cells so that we can build a lab on a chip device that can say you have this much percentage of cell A, cell B, cell D. And if we can do that, we can also use the device for drug discovery purposes as well. So that covers actually 4 faculty members. Is there anyone in the ME? I cannot think about anybody else. Of course, in ECE, Bruce Gunade, if you're aware of him, has ongoing interesting research. Maybe Malcolm McFarlane, as well as JC Chiao. These are all people that you can reach out and talk to.
Thank you.
I'm sorry that I responded in a very long way, but perhaps it will guide some of you towards the correct faculty members. Any other questions?
I have a few.
Yes, please.
I'm curious to know what the percentage accuracy is of the test, because I know currently most antibody tests aren't very accurate. And if I remember correctly, the threshold that's required for emergency FDA approval for antibody tests is only 50 or 60% accuracy. So I'm curious to know.
Oh, that's a good question. So these results that I show here are highly repeatable. So that's the important thing about it. So the signal is about 20% in the lowest concentration that we have done. So what we are hoping to do is really do a systematic study this summer and reduce the amount of basically antibodies that we're going to introduce and systematically see, what we really measure. Are really measuring it with the desired specificity and sensitivity. So you need to actually look into these. And these, unfortunately, or fortunately, is a long-term study that you have to do basically in the lab. So I'm sharing with you some very early results, but these results are highly repeatable. So I'm hoping that, we will be able to go through the FDA approval if we have a strong data showing this.
Wonderful.
Question, indeed, thank you.
Dr. Beskok?
Yes.
I have a question. Is it necessary to test all the different antibodies at different times, or could you just pull them all into one and test them all for, all 3 or all 4 at the same time?
I mean, at this point, I'm testing, all 3 at the same time, and the other one's a negative control reservoir. So this is for, you have to have positive and negative control in your test. But your question is very relevant because we know that there are different variations of COVID-19, right? So what makes that variation is changes in the spike proteins of COVID. So if I actually, have access to this, I can redesign this, not only to go IgG, because IgG is what you need for long-term, immunity against, COVID-19. All you need is IgG. You don't need IgM or IgA. But the idea that I've shown you happened at the beginning of the pandemic. So first IgM came. So if you could actually measure IgM fast and accurate, maybe you can say, oh, you've been exposed to COVID, your body's building these. So that's why we put these over there. But, in another revision, we can actually put different spike proteins of different variations of it, so that we can actually say, oh, you have South African or you have British, variation, which will be important too, because then, that you build immunity towards those as well. I hope that answers your question.
Yeah, great. Thank you.
Any other questions? Okay. Well, I'm going to, tell the JC that the rest of his group did not come. But thank you for coming, indeed. It's been a pleasure.